There are disorders where the neurons don't migrate properly. Some of those patients are really smart. They're like totally normal. They can be medical students, they can be CEO. I've seen people that are CEOs of companies, computer programmers, like way smarter than I am. And they have all these neurons in the middle of nowhere. And to our wonderment, we found that the genome in every neuron is different from the genome in the neuron next to it. And that it's a dynamically changing thing. That each one of our neurons accumulates about 20 mutations every year, like a clock. These mutations measure our age with incredible precision. And then we find that if you have a degenerative disease like Alzheimer's disease or ALS, Lou Gehrig's disease, all of these conditions that we've studied so far, the neurons develop extra mutations.
Has this entire narrative been complicated a little bit by the discovery of neurogenesis, or the proliferation of neurons throughout life? Have you ever wondered how a 3-pound organ containing billions of neurons manages to build itself? Why do some children develop neurological conditions like epilepsy or autism while others don't? And what if the genetic mutations that influence brain disorders aren't always inherited, but can arise during brain development itself? For decades, scientists thought of our DNA as a fixed blueprint that remains largely unchanged throughout life. But what if every neuron in your brain carries its own unique genetic story? Today's research is revealing that brain development is far more dynamic than we once imagined. From the earliest stem cells in the embryo to the mature neurons that support thought, memory, and behavior, tiny genetic changes can shape how the brain develops and sometimes contribute to neurological disease. Hello, I'm Avnish, and this is The Early Perspective, a podcast where we explore how the brain works, how it changes, and what that means for how we learn, think, and live. Today, we're going to be diving into one of the most fundamental questions in neuroscience. How does the human brain develop? We'll explore how genes guide the construction of the cerebral cortex, what happens when development goes off course, and how genetic mutations arising throughout life may influence conditions ranging from epilepsy and autism to Alzheimer's disease and dementia. To help break all of this down, I'm incredibly excited to welcome today's guest, Dr. Christopher Walsh. Dr. Walsh is the Chief of Genetics and Genomics at the Boston Children's Hospital, an investigator of the Howard Hughes Medical Institute, and a professor of pediatrics and neurology at Harvard Medical School. He earned both his MD and his PhD from the University of Chicago, completed his neurology residency. He earned both his MD and his PhD from the University of Chicago, completed his neurology residency at the Massachusetts General Hospital, and later trained in genetics at Harvard. His research uses genetics to understand how the human cerebral cortex develops and how disruptions in that process leads to neurological disorders. His lab has identified dozens of genes involved in brain development and linked them to conditions including epilepsy, intellectual disability, and autism spectrum disorders. More recently, his work has shown that mutations arising during development can contribute to disorders such as epilepsy, autism, and schizophrenia, and that individual neurons accumulate unique mutations throughout life, even after they stop dividing. These discoveries are helping reshape how scientists think about the brain and how it develops. Dr. Walsh has been elected to the National Academy of Medicine, the National Academy of Sciences, and the American Academy of Arts and Sciences, and he was also awarded the Gruber Neuroscience Prize in 2021 and the Kavli Neuroscience Prize in 2022. Dr. Walsh, thank you so much for joining me today.
Thanks, Avnish. I'm really glad to be here. It's always fun to talk about the brain.
Of course. Okay, so to start off, I'd love to go back to the beginning. Could you tell us a little bit about your journey into neuroscience, medicine, and just brain development research in general?
Sure. I think in my case, my decision to go into neuroscience is really influenced by my teachers. I skipped my last year of high school and ended up in college just a month after I turned 17 with no idea what I was going to do or what I was going to study. And I took 2 courses my first semester of freshman year that really have defined my trajectory ever since. One was psychology, and one was organic chemistry. And ever since then, I've been fascinated by this interaction between chemistry and the brain. And that got me on the road to neuroscience research that I, continued to follow in college and then in graduate school. And so I'm really grateful for those teachers that really inspired me, which is part of why I love to talk to people who are at a younger stage in their career than me.
How was that process, that transition from just skipping a year of high school to college straight?
Well, that happened because I come from a big Irish Catholic family.
Mhm.
I'm the seventh of eight kids, and we, we were comfortable, but we didn't have a lot of money, so everybody had to save money on college somehow. And I was the only one who didn't go to Catholic school, so I was actually going to a private school, and so my parents left the choice up to me. They said, you can skip a year of high school or you can skip a year of college like your older sister did. And I didn't particularly want to skip a year of either, but college seemed like it might be more fun than high school. And so that's how I ended up in college after 3 years of high school. And like I said, I really wasn't thinking about doing that. So compared to how much kids are prepared now for college and high school, I was like completely unprepared to even have any idea. I entered with no major and no really idea what I was going to do for a major. So if
Mhm.
any of your listeners feel like, they aren't on enough of a path, I'm here to say you don't have to know what you're doing. You can figure it out as you go.
Yeah, that's honestly wonderful advice. So, what first drew you towards specifically the developing brain?
Well, that's a good question. And I'm not exactly sure why. I guess that was mostly in graduate school.
Hmm.
When I first went to graduate school, I wanted to work on schizophrenia because it illustrates this interaction of chemistry, namely dopamine, and behavior. Uh, because even way back then, it was known that dopamine was important. But at my school, there were a lot of great people doing great science, but nobody was doing great science in the area of schizophrenia. And so then I was in a um, little bit of a crisis. I didn't know what I was going to work on. And I just started again talking to a bunch of different professors, kind of trial and error. And I must have talked to 15 different professors about their research. And so I met with Professor Ray Guillory, uh, who was working on the development of the brain and in specifically genetic mutations that disrupt the development of the brain. And he just immediately inspired me about the fact that the brain is like so specialized, but it just starts out as a bunch of simple cells that look just like any other cells, and then somehow this miracle happens that gives us this ability for consciousness and
Hmm.
free will and choice and intellect and creativity. And so I've always been fascinated by how that miracle comes about.
Yeah, of course, it's an incredibly fascinating topic to get into. So, when you did start going into that topic, was there any particular discovery or experience that shaped your career and the future trajectory?
Well, again, there are lots of things that have shaped my trajectory. I sometimes feel like I'm a blank slate and I just am imprinted by the people I encounter. I had some really good research experiences in college that helped me along, but my, again, it's been a series of things. I mean, really, my role model was Ray, my
Hmm.
PhD advisor. He showed me what it was like to do science, how it could be just a really fun thing, a little bit like art, except it's also a dispassionate search for truth. And
Right.
so I just love the fact that you can be creative, but you're bound by rules of trying to find something which is true and durable. And, just trying to, you know, help expand knowledge a little bit. And I had a very dramatic experience my first year of graduate school uh, because I discovered along the way the idea of doing a combined MD PhD degree,
Hmm.
and that was a moment for me. I again, I wasn't sure if I wanted to go to medical school or graduate school. I felt like I liked science more than I was enjoying my volunteering in hospitals, because in the science lab, anybody can do anything at any age. And so I was given a lot of responsibility, whereas in the hospital as an undergraduate, you just kind of follow other people around.
Right.
And so I was wrestling a little bit with my decision between medicine and science. And one day I walked into my pre-med advisor's office and I saw this pile of pamphlets on the side. And I still remember exactly where they were and where his bookcase was. And I absentmindedly picked up one of these pamphlets. And I'll never forget what it felt like. It's like this high quality sort of textured paper, and it has this burgundy color. And that was a pamphlet describing the combined MD PhD program at Cornell Rockefeller. And I must have read that pamphlet about 100 times over. And that was a moment where a light bulb went off for me, that this was the way of doing this. And they actually would pay you to do this, that it was free. And again, coming from a big family where we had to think about saving money, it was a perfect thing from my family perspective also. But and so, that was really a moment that set me on the course to doing the combined degree programs.
Right. So then, once you did go to medical school and also graduate school combined, how did you get to choosing neurology as your specialty? What guided your decisions in medicine?
Well, so I really enjoyed my PhD in neuroscience. And so I decided I wanted to stick with neuroscience research. And, the first 2 years of medical school, you're mostly in the classroom. And then I stepped away from medical school to do my PhD. And then I was thinking I would probably want to do neurology as a specialty, but wasn't certain. And from there, you basically just try different specialties as rotations. You do a month of neurology, you do a month or 2 months of internal medicine, and it's really very empirical. I've talked to a lot of medical students about this choice because I was director of Harvard's MD PhD program for a few years. And I think of it as like the wand chooses the wizard, because you walk into a rotation and there's some where you just feel comfortable. You just feel like you're at home. And there's others that you thought you might like, but they just don't feel right.
They don't
And
click,
so it was,
yeah.
yeah. And so it's really on the one hand, it's a gut feeling. On the other hand, as the seventh of 8 kids, I've always looked to role models. And I told you that Ray, my PhD advisor, was a role model because, it looked like he just had a life that I could enjoy living. He worked in the lab, he chatted, he stopped for a cup of coffee, he'd go home, he'd have dinner with his family. And I knew that family was going to be important to me and that I'd want to have a family of my own. And then, he'd do reading and things like that in the evening. So he seemed like he had a life that was a good lifestyle, as well as a lot of fun. And so when I was doing those rotations, I looked around at the professors who were in the medical specialties, the clinical specialties. And again, I could see myself identifying with the neurologists who, again, did a lot of research because we don't have cures for a lot of neurological diseases. So research is very important.
Right.
And so that's valued by that specialty. But they also had a lifestyle that looked like it was going to be, one that I could enjoy.
And then has working with patients in general influenced the questions that you ask in your research?
Absolutely. And that's one nice thing about doing medically related research is that I derive a lot of sense of purpose and meaning from doing research that matters to people. I think it's a general question in our lives: how do we find purpose and meaning? And for me, I like the idea of discovery, that I'm somehow advancing knowledge, but I also like the feeling that those discoveries can help people. And so when I first started my lab, I thought I was going to do really basic stuff on like rats and mice. That's what I'd been doing for my postdoctoral fellowship. I was studying how the cerebral cortex develops from these undifferentiated cells, marking stem cells and looking at the clones that they would form and how they would migrate to the cerebral cortex. but I kind of wanted to do something more related to patients because I was, after all, a neurologist at that time and wasn't sure exactly how I was going to do that. And after I'd started my own lab, I went to a meeting to present my work. And one of my teachers from the University of Chicago, the pediatric neurologist by the name of Peter Huttenlocher, presented a family with an inherited malformation of the cerebral cortex. And again, that was one of those aha moments for me when I realized that this is exactly what I want to do. This is a way of applying genetics to the study of the development of the brain, and a way of doing it in a way that's going to give us durable information, that will always be true. Like if we find what that gene is that's abnormal in Peter's family, that's always going to be true science. That's not going to be something which a new technology will come along and find was actually wrong. Because this is a problem in science, that a lot of things we discover, 1 year turn out to not really be true or useful a couple of years later. And I find it important to try to do something that will be durable.
So, do you think that doctors and scientists think about problems differently, or do you think they take the same approach and apply it in just different fields, I guess?
That's a great question. And doctors and scientists think about things very differently. There's a completely different theory of knowledge or epistemology between the medical side of taking care of patients and the research laboratory. And that's one of the challenges of the combined MD PhD program, is because I came out of my PhD thinking I knew something, and I entered my final 2 years of clinical training thinking I knew something, and I knew nothing about how to take care of patients or how the medical mind worked. And so, because a lot of it has to do with that interaction with patients, and it's very empirical, and there's things that don't always necessarily make scientific sense that just work. Whereas in the lab, you can reduce things down to a defined experiment and a hypothesis. On the other hand, physicians who do research, they follow pretty much the same theory of knowledge and the same pattern of experimentation as, PhDs who do research. And so, you really can't, at the highest quality of research, you can't tell, whether someone's coming from a medical background or a straight science background, if the work is good.
Right. Yeah. So then you're saying, of course, there are a lot of differences, but ultimately could be similar. They might take a similar philosophy of approaching things, right?
Exactly. Right.
And then just to pivot, since we're talking about your research, I just want to get into that specifically. For many people, it might seem that it's almost impossible that the brain is building itself. So starting from a fertilized egg, how does the cerebral cortex actually form?
Sure. Well, the early steps of forming the brain are, generally similar to the formation of other organs like your heart or your gut. It has to be patterned, and there's a whole science field about how, the body knows that this is the head end and that's the tail end. And how the inside is formed versus the outside. So there's a lot of early patterning events that are kind of shared by the different organs. And then all of the different organs have stem cells, and they have to make, these are the grandmother cells that are undifferentiated, that divide and produce more cells. And that's pretty similar in the different organs as well. And then, cells are sometimes formed in places where the stem cells lived, and sometimes they have to move or migrate to where they differentiate. And that happens a lot in the cerebral cortex. For some reason, the stem cells of the cerebral cortex are buried deep inside in the inner lining of the brain. And it forms the neurons, which don't divide. And then after the neurons have withdrawn from the cell cycle, they migrate out to the outer surface of the cerebral cortex, where they stop dividing, and then they start forming connections. And so that's, again, somewhat similar to other organs. But then after that is where the brain gets really interesting, because then what happens is the different brain cells, particularly the neurons, start talking to each other. And they regulate a lot of different things. The neurons that are formed start talking to the stem cells and start controlling how those stem cells proliferate. And then the neurons start forming connections with each other. And that, in fact, the neurons we've recently discovered, they undergo a Darwinian struggle for survival. They're formed in excess by about 30%, and they compete to survive. And the ones that survive are the ones that successfully form connections called synapses with other neurons. And so it's this, like I say, it's this really competitive process right around the midpoint of fetal gestation in humans, where these neurons are competing like crazy. And then, of course, these connections continue to be elaborated throughout our whole life. That's what learning and memory is all about.
Right.
But this aspect of the interactions between cells is under a less strict sort of genetic control. And so the early steps of the formation of the brain are under mostly genetic control, similar to the early development of the other organs. But then the brain has this extra level of this interaction and this selection and this activity-based development that's fairly unique to the brain. And that's where a lot of the magic is. And so although there are a lot of genetic disorders of the brain, we have a lot of the development of the brain is also non-genetic and is patterned by this sequential series of interactions.
Right. So there is a sequential process of how it's developing, right?
Exactly, yeah.
What's very interesting is, I guess, stem cells itself. These cells are, I guess, undifferentiated and almost like a blueprint because they can become into other things. But how does that process work? Because these are the most versatile cells that we have, basically, right?
That's right, yeah. So again, like I said, the early steps are fairly strictly controlled by genes. And so, for example, there are genetic disorders in humans where the stem cells just stop making neurons too soon. And so the brain ends up too small. So our brain might be reduced to the size of a chimpanzee's brain. So that's not an uncommon genetic disorder of kids. Or there are disorders where the neurons don't migrate properly. So they might migrate, instead of all the way to the cortex, they migrate halfway and then for some reason stop in the middle of nowhere. So these early steps, like I say, are fairly strictly genetically regulated. And there's a host of hundreds of thousands of different rare genetic syndromes in humans, each one individually very rare, that disrupt it. But then, like I say, the neurons get to where they're going and they start making connections. Then there's a whole other series of conditions in humans that disrupt the way the connections are made and reinforced. And so in those connections, the brain might look pretty normal if you took an X-ray or an MRI scan, but it just doesn't work properly. And those kids will often have intellectual disabilities or autism spectrum disorders, because even though they've got the right numbers of neurons and they're in the right places, they just don't connect properly.
Right. So first it's primarily just genes influencing, and then it's another whole host of other factors which influence the development, right?
That's right, exactly. And these later steps are influenced progressively more and more heavily by what we experience.
Right. And then another major part of your research has been cortical malformations and understanding what it is.
Mm-hmm.
So, of course, that seems really complicated, but what exactly are cortical malformations?
Well, so cortical malformation is more or less the way it sounds. It's the brain just doesn't get put together properly. And we diagnose them by putting a patient into an MRI scanner. And an MRI scanner can take a three-dimensional picture of your brain, non-invasively. And so we can see with tremendous detail now the inner structure of the human brain. And so we can characterize these sometimes even subtle disorders of the way the brain gets put together, using the MRI scan. And then we can connect that to the genetics.
And then how exactly do they arise and during development?
So genetic mutations, they can be inherited, or they can be spontaneous mutations. And so when it comes to things that affect the brain, they often are associated with social challenges or intellectual challenges. And so people that have disorders of the brain will often not have a family themselves. They might not, be smart enough to be able to manage it, to get married and have kids. And so given that, the disorders that affect the brain that are inherited are often inherited in what we call a recessive fashion, where you have to have disruption of both copies of a gene. So just to back up for a minute, most of our genes we have two copies of. We get a copy of every gene from our mom, we get a copy of every gene from our dad, except for the X chromosome, where females have two copies and then males have one X and one Y, of course. And so for most of the genes in our genome, one copy of the gene is all we need. And so we can actually have a disabled copy of one of the two copies of a gene and not know it.
Right.
And it only is a problem if an unfortunate child inherits a disabled copy of the same gene from both parents. And that's what a recessive disorder is about. And like I say, most of the disorders then of the brain that are inherited in this recessive fashion, where the parents didn't even know they were carriers, or these days sometimes people will get testing. And it's just bad luck that the disabled copy from both parents goes to the same child. But in fact, a lot of the genes that disrupt the development of the cerebral cortex or the function of the cerebral cortex are not inherited at all. They're spontaneous mutations that affect just a single sperm, or less commonly a single egg of one of the two parents. And they affect a gene where you need both copies, because for some reason the brain needs both copies of the gene. We call those dosage sensitive. It's something that causes a condition even if you're just missing one of the two copies. And so these are in theory would be inherited in dominant fashion. If for those of your listeners who've learned about dominant inheritance and recessive inheritance, but again, they're not usually inherited because of the fact that they're so disabling. And so we realize now that these mutations that disrupt just one of the two copies of the genes are only appearing in our population because new mutations are constantly happening. And so it turns out that each of us has about 80 to 100 new mutations relative to our parents. Fortunately, most of those aren't in the middle of a gene or disabling to the gene. Most of them are in the junk DNA that makes up, 95 or more % of our genome. But in some unfortunate cases, there can be a spontaneous mutation, just a bolt from the blue. Nobody knew it was coming, that affects an essential gene. And that's a common cause of cortical malformations or autism and intellectual disabilities and epilepsies as well.
Right. So you're saying that the genetic component could be something inherited from parents who are carriers, and they have they have a recessive copy of the gene and they don't even know it. And then
Right.
that, unfortunately, the two recessive copies leads to the disorder. But then there could also be mutations itself arising randomly, and that could ultimately lead to a dominant kind of fashion of it showing up, right?
Exactly. And once the dominant mutation occurs, in theory, it could be inherited. If the affected child grows up and has kids, they would then transmit it because they have that mutation in their,
Right.
in their sperm or in their eggs. But again, those kids are often unable to have a family themselves, and so therefore the mutation is not transmitted. And so it really only appears in children because of this constant creation of new mutations in our sperm and egg cells.
And how do these seemingly small scale mutations lead to large scale developmental disorders like epilepsy, autism, or schizophrenia?
Well, that's a complicated question. I'll try to give a few examples to try to make it simpler. One way is in the case of epilepsy. So epilepsy is uncontrolled electrical activity of the brain. It can cause epileptic seizures that are, repetitive movements, usually associated with loss of consciousness and control. And so that those epilepsy mutations are frequently mutations in the ion channels that regulate the excitability of neurons. And so it disrupts the pattern of electrical activity of the neurons, and periodically that can become unleashed to drive a seizure. In the case of intellectual disabilities or autism spectrum disorders, again, there the genes are often the genes of the synapse that are actually required for neurons to connect to one another. And they're the genes that are active when we're trying to learn something. Those genes are upregulated or altered in their pattern of expression. Again, missing then one of the two copies of that gene can disrupt that whole process of learning, which is what results in intellectual disabilities.
I see. So then these mutations are affecting larger scale processes which end up causing these disorders in a way, right?
Exactly right. Yeah.
Okay. And then, of course, your lab has identified dozens of genes that are involved in human brain development. So how do researchers go from studying families with neurological disorders to discovering entirely new genes?
Well, that's been that approach has evolved dramatically over the time that I've been doing science. We've now had, we just celebrated the 25th anniversary of the sequencing of the human genome. And that was a revolution in terms of our ability to find new genes and assign a function and a disease to those genes. And so, you could see how like we are discovering genes at this slow rate. And then all of a sudden it just like accelerated exponentially after the sequencing of the genome. Now it's sort of leveling off because with the sequencing of the whole genome, now the way we discover new genes is we see a kid in the clinic and it's become a routine clinical test when it's indicated to sequence the genome of the child. And we always have to compare that genome to the genome of the parents. We usually will find a gene that's previously characterized as causing that child's condition, but sometimes we'll find, a mutation in a gene that hasn't previously been described to cause intellectual disability or epilepsy or autism. And then we call up colleagues who are doing the same thing. And, I've also seen another family in another part of the world that also had an unexplained mutation in that same gene. And then we know that it's a new gene for a new disease.
Yeah, that's a very interesting approach to it. But then you said that it's drastically evolved since, I guess, just a couple decades ago. So
Yeah.
how's this process like when you first started?
Well, when I first started, we didn't really have a map of the genome. We didn't know where any of the genes were. We knew that how many chromosomes there were and stuff like that, but we didn't really have the beginnings of a sequence. And so when we cloned our first couple of genes, we actually had to identify, we narrowed down the gene to a certain portion of the genome, and we knew it was maybe 5 or 10 million letter. The genome, of course, is just 4 letters, A, T, C, and G, and we narrowed it down to about a segment of maybe 10 million letters long, which probably had like a couple of dozen or maybe 100 genes in it, but we didn't know what those 100 genes were. And so we had to just start like sequencing that place and try to figure out what are the genes in there. And so, the first gene that we ended up identifying is called double cortin because it disrupts the migration of the neurons. And so some of the neurons migrate halfway to the cortex. And so instead of having one cortex, you have 2 cortexes, an outer one and an inner one. When we discovered that gene, it was not known to be a gene. It was just like a random piece of DNA that hadn't really been put together. And so that's what it was like when we first started back in the 1990s. And like I say, then around 2000, the genome was sequenced. And then a couple of years later, there were ways of basically just sequencing at least the coding parts of the genome in every patient. So we didn't have to map the gene anymore. We just would go right to sequencing every gene in the person's, all 20,000 genes in the genome in that person. And so that's how we do it now. You just basically, you don't have to do any mapping, no cloning. You just go right to sequencing every single gene in the genome. And we know so much now about the patterns of variation of the genome in normal people that it's very easy to recognize a variant which doesn't obey the, which is out of the bounds of the normal variation.
Right. So technology has revolutionized the process, made it much more efficient, and I guess given a lot more knowledge and ability to physicians and scientists alike, right?
That's right. And now it's, like I say, now it's not really a job for research labs anymore. It's now a clinical test.
Yeah.
And it's the clinical labs that are, continuing to discover new genes, which is great because now this is a service we can offer our patients.
Mm-hmm. Yeah. And then, of course, you've studied a lot of these disorders and conditions, but what has that taught you about normal brain development in general?
Well, I think it's taught me a bit of what I was describing about this sequence of events that regulates how the brain develops. This fact that the early steps are governed more strictly by genes and the later steps are more governed by interactions. That was something that I have a much greater appreciation for now than when I first started. It's taught me just some amazing things about just how variable the development of the human brain can be, and then somebody can still be basically normal. And so the first gene we started studying, a subset of the neurons never migrate at all, and they form these blobs of neurons way deep in the brain, nowhere close to where the cerebral cortex is supposed to be.
Mm-hmm.
And we identify those patients because most of those patients will have epilepsy, although sometimes the epilepsy is pretty mild. But some of those patients are really smart. They're like totally normal. They can be medical students. They can be CEO. I've seen people that are CEOs of companies, computer programmers, like way smarter than I am, and like actually have all these neurons in the middle of nowhere. And it's kind of a mystery to me whether those neurons that are all out of place, do they actually wire up to the rest of the brain? Like, are people thinking with those neurons that are in these irregular blobs? Because the people like me who are obsessed with the brain, we're obsessed with the beauty of the organization of the neurons in the cerebral cortex. They form these 6 layers like a layer cake, and they have this beautiful shape and morphology and these beautiful long connections.
Mm-hmm.
And we think, and it's natural to assume that this beautiful organization is essential for its normal function. And yet I see patients who've got cells all over the place, and they seem to be, some of them are doing just fine. Not all of them. They have a higher rate at which they might have, cognitive challenges, but some of them are amazing. So I'm really impressed with just how diverse a normal human brain can be.
Right.
And that there's no like one path, that the brain has to follow in order to develop. And that, genetics is important, but it's by no means the whole deal.
Right. Yeah. And then since we're talking about how, I guess, diverse a normal brain could actually look like, what exactly does determine whether a mutation or a malformation is clinically significant? Because, of course, a lot of it could be there, but that might not even show up, right?
Right. Well, I think I think there are a lot of things that regulate that, and we don't understand most of them. This is a phenomenon in genetics that's referred to as variable penetrance. And this is true of all genetic disorders, that they show variable penetrance. And in fact, the more we sequence the genome of normal people, the more we find that some of them like have mutations where they ought to have a disease, but don't. Or they have some very mild form of a disease. For example, cystic fibrosis is a very severe disease of kids where they can't breathe properly. They get pneumonia. They typically die by the age of 20 or so. They have a lot of trouble absorbing food, so they're malnourished. And so when I was a medical student, this was a uniformly fatal diagnosis, or so we thought. Now with genome sequencing, we find that certainly some kids with cystic fibrosis have that severe course, although now we have drugs that can treat that thanks to harnessing the genetics of cystic fibrosis. However, we also discover that some people have like those exact same mutations and they don't have any of the lung problems or any of the gut problems. And that's a mystery, exactly how that happens. And that's very true in the brain, where particularly girls are protected from mutations that affect the brain. And we see this most drastically in autism spectrum disorders, where autism is 4 or 5 times more common in boys than girls. And that's been a bit of a mystery, but it's something we've been studying a lot lately. And we think we have some thoughts about what might be regulating that. It's very poorly understood, this phenomenon of variable penetrance. It might have to do with the genetic background. It's on part determined by the other genes that people have, because genes kind of interact in complex networks. It's probably also regulated in part by random mutations that we all develop as our bodies develop, since we all have patches of cells that are abnormal. And that's a kind of a somewhat random process. And then it may, and it's probably governed by other things that we don't understand yet.
Yeah, that's incredibly interesting because I never realized that gender could really influence, I guess, the number of people who had disease. Like 4 or 5 times is a huge difference. So how much do we understand? Is it just mostly an understanding of the factors, but have we pinned down any real conclusion from that?
Well, so, there are certain diseases that are more common in girls than boys, and then there are other conditions that are more common. Sorry, there are certain conditions more common in girls than boys. There are other conditions more common in boys than girls. It's more frequent that things affect males more than females. Things that affect females are more commonly diseases of adulthood and things that affect the immune system, things like lupus, systemic lupus erythematosus, multiple sclerosis. A lot of autoimmune diseases tend to be more common in females than males. And it's not totally clear why. But then it's often the developmental conditions that affect boys more commonly than girls. And it's not just developmental conditions related to the brain, but it's very prominently developmental conditions related to the brain. That includes not just autism spectrum disorders, but attention deficit, hyperactivity disorder, schizophrenia, but then also developmental disorders of other tissues. So also developmental conditions that affect our gut and our heart. Congenital heart disease, for some reason, is 1.5 to 2 times more common in boys than in girls. And so this probably relates to something about how the sex chromosomes interact with the rest of the genome. So again, girls have 2 copies of the X chromosome. Boys have an X and a Y. The Y is a very tiny chromosome that has very few active genes on it. And so girls actually have, 2 copies of a lot of genes. So they have the potential for higher expression of some of those genes. And it may be that our going theory is that there's something about those 2 X chromosomes that tends to protect females from a lot of childhood diseases.
Right. So the interaction of the sex chromosomes is allowing potentially for this kind of protection, right?
Right. For example, there are genes on the X chromosome that encode regulators of other genes. They're genes that encode proteins called transcription factors, which regulate how a gene somewhere else in the genome is turned on or turned off. And so they may, particularly for these diseases where you need both copies of the gene to work, they seem to maybe boost the expression of that gene elsewhere in the genome, and boosting the expression of a little bit might protect someone from losing one of the 2 copies.
Right, I see. And then, since we're on the topic of disorders, have we seen their pathways overlap? Because I know we previously discussed that a lot of it is caused by mutations of a specific system, and that's like kind of causing a chain reaction. But then, are there overlaps in that system or pathway?
Yeah, well, there's a lot of overlap in terms of the genes associated with autism spectrum disorders. They highlight, again, mysterious condition because it also can be caused by hundreds of different genes, can cause a condition which, when you see the child in the clinic, is indistinguishable. These 100 different kids can look the same, but they can have different genetic mutations.
It's all causing the same thing, yeah.
Yeah, and that's different than the way genetics usually works. Most of the time, there might only be one or two or maybe a dozen different genes that cause a particular condition. It's rare to have this level of diverse different genes that can cause a seemingly indistinguishable condition. That seems to reflect the fact, though, that a lot of these genes function at these connection points between one neuron and another called synapses. And those synapses are just really complicated structures. And they have about 1,000 different proteins in them. And all of those proteins look like they're really important. And so it looks like you knock out any one of them and there's something not quite right about the synapse. And if the synapses aren't quite right, then you have difficulty learning things, and you have difficulty learning social interactions, which is some of the most challenging things actually that humans have to learn. One of the hardest things to learn is how to interact with other people. We learn that long after we learn to walk. We learn that after we learn to talk. It's only like in kindergarten that we really start learning how to interact with people and how to detect social cues. And because that's a really hard thing for the brain to do.
Right, I see. Well, I also wanted to pivot into one final topic before we close for today. So I know your research also suggests that mutations accumulate faster in neurodegenerative diseases. So how might that connect to conditions like Alzheimer's disease or dementia?
Right. Well, so we're very interested in how that connects. And so, this has been some findings that have really kind of been mind-blowing to me because, I've been in neuroscience for a long time. I'm an old person. And all that time, I just thought that, neurons are not dividing cells, and nature made them to be non-dividing so that their genome would be protected and it would stay the same their whole life. Unlike other cells like skin cells and, blood cells that are dividing all the time. And we know that they can sometimes form cancers. But neurons in the brain, since they don't divide, they never form cancers. And we thought their genomes were very stable. And so we started sequencing the genome of a single neuron by amplifying it, taking one neuron and putting it into a well and isolating its genome and then sequencing it. And to our wonderment, we found that the genome in every neuron is different from the genome in the neuron next to it, and that it's a dynamically changing thing, that each one of our neurons accumulates about 20 mutations every year, like a clock. These mutations measure our age with incredible precision. And if we live to be 100, we accumulate 20 mutations the first year of life in each one of our neurons and 20 years of our 100th year of life in those neurons if we don't have any disease. And then we find that if you have a degenerative disease like Alzheimer's disease or ALS, Lou Gehrig's disease, all of these conditions that we've studied so far, the neurons develop extra mutations. And it looks like those extra mutations are caused by the disease because the pattern in which the mutations occur, the particular number letter substitutions are different. The ones that accumulate with age are generally changing a C to a T or changing a T to a C. But in Alzheimer's disease, we most commonly see that now we're changing a C to an A, and that's a change which almost never happens in a normal neuron. And that's something which is probably caused by the disease, and it's probably caused by the fact that the brain is inflamed, that the immune cells of the brain called microglia are a little out of control. And they're secreting things that are probably damaging to the neurons.
So then these mutations could potentially contribute to direct neuronal damage, you said?
We think that they do. We think that the ones that occur in normal aging, even though it's 20 per year, and even though it's, thousands by the time you're my age, that they don't seem to be necessarily terribly damaging to the way the neurons work. But anything that increases that more than a little bit can be very damaging. So in fact, some of the patterns of damage we see in Alzheimer's disease, a neuron would have an excess of mutations equivalent to more than 100 years of normal aging. And so that's probably very damaging to the function of the neurons.
And then has this entire narrative been complicated a little bit by the discovery of neurogenesis, or like, I guess, the proliferation of neurons throughout life?
So yeah, the proliferation of neurons throughout life is a fascinating topic. And that it does not happen so much in the in the neocortex of the cerebral cortex. So the biggest part of our cerebral cortex is a part that's called the neocortex or new cortex that appeared relatively recently in evolution. There's another part of the cerebral cortex called the hippocampus, which is an essential center for memory, and that is much more highly conserved over evolution. And so we see it in a lot of animals that we diverged from a much longer time ago. So it's called the archicortex or the old cortex. So that ongoing formation of neurons looks like it happens in that hippocampus, in that old cortex. And that's a fascinating process, both in terms of how it might regulate normal learning and memory and aging, but also we're finding some connections to age-related diseases that might relate to that process as well.
Mm-hmm. Yeah, I see. Okay, so before we wrap up for today, I just wanted to do a quick rapid-fire round of questions.
Okay.
I just want you to give me your honest, quick answer to each of these questions.
Okay, great.
Okay. So, what do you think are the biggest unanswered questions in brain development research today?
Well, I think the biggest one is like, what is memory? Like, how do we remember things? We know that it has to do with changes in these connections called synapses, but exactly how that happens, still a huge mystery.
Still unknown, yeah.
Yeah. There's a lot, we still don't know a lot about this disorder, schizophrenia, that I went into neuroscience wanting to study. Many, many decades later, we still don't understand a lot about exactly how can it be that people start hearing voices? So those are just a couple of big questions. There's lots, I could talk about them all day.
And then what do you think is something that researchers are getting close to understanding that once seemed impossible to understand?
Well, I think one thing we're getting close to understanding is the genetic basis, particularly of pediatric disease. The genetic basis of a lot of diseases we understand pretty well. You can say we're close to understanding it, or you can say we're far from understanding it. For most conditions, if we have a kid that comes into clinic and we think, oh, he's probably got a genetic condition or she's got a genetic condition, and we sequence their genome, we have about a 1 out of 3 chance of finding the genetic basis of that. So compared to when I started, that's really, really good. Compared to where we'd like to be, that's still really bad. What's that other 2/3? So, that's both something that we understand well and something we understand poorly at the same time. So we really have a lot. We are, as I say, we're starting to level off in our discovery of new disease genes. And so we're starting to saturate that. And so we're starting to have a pretty good sense of the annotation of our genome. But there's still lots of families out there that still need our help to understand what's the matter with their child.
And then for students who are listening and they want to go into neuroscience, medicine, genetics, or just developmental biology, what advice would you give to them?
Well, I think the best advice is to try to see if you can find a way to give it a shot working in a laboratory. If you're near a big city or if you're near a university, the chances are pretty good that if you reach out to some of the laboratories there and ask them, there's a good chance you might be able to, volunteer over a summer or something like that. And I think it's important to try that because, like I say, for me, I didn't know what I wanted to do. And I only learned what I wanted to do by trying stuff because we don't really know ourselves that well. And we don't know really what we're going to like and what we're not going to like. And the only way of finding if you're going to like something is to just kind of like walk in and give it a shot. But also, I think it's important to try to see if you can find a connection to a lab, because people think of scientists as cloistered in these white coats and being, really formal and structured. And science is exactly the opposite. It's something where everybody's like wearing t-shirts to work and they come and go as they please. And they're trying to really be creative. And so it's really different than the way scientists are portrayed.
Of course. And then, since we're talking about how there is a misconception about science, what do you think is a misconception about being a physician?
Well, I think, and also one thing that people don't realize about physicians is that uh, there are tremendous range of career choices for a physician, other than just being a physician. Being a physician is tough these days with the insurance companies and things like that. There's a lot of burnout among physicians, but there's all sorts of career paths you can take after medical training. And so it's very empowering. And the other thing about, medicine is just that it's just an incredible privilege to be a physician and to have the opportunity to talk to people at their time of greatest need and to feel like you might be able to, play some small part in helping out.
Right. And then the last question I want to ask you today is, if listeners could only remember one thing from this entire episode, what would you want that to be?
Gosh. Well, I think it would be something like, just science is fun. Science is like really different than people think of it. It's a really creative thing. Uh, and, and and like I say, it's a fun life. So I highly recommend it.
Okay, great. Dr. Walsh, thank you so much for joining me today and for helping us better understand one of the most remarkable processes in biology.
Well, thanks very much, Avanis. It's been a real pleasure.
Yep, of course. The human brain doesn't simply appear, but it is built cell by cell and through an intricate developmental process shaped by genetics, experience, and just time. And by studying how that process unfolds, scientists are able to discover new insights into disorders ranging from epilepsy and autism to Alzheimer's disease and dementia. For listeners, if this episode gave you a new perspective on how the brain develops, be sure to share it with somebody who you think would enjoy the conversation. If you guys enjoyed this episode and learned something new, make sure to follow on your favorite podcasting platform or on YouTube. I'm Avnish, this is The Early Perspective, and I'll see you in the next episode.